Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we have already started with a lecture series of biological classification chapter of class 11th. And in that, we have already discussed about the different classification systems, kingdom Monera and kingdom Protista. In this lecture, we are going to study the detailed, thorough study of this kingdom fungi. So first, by listening to this term fungi, we need to imagine where these fungi are found, which type of organism are known as fungi. Dear students, sometimes in our houses, if we leave a food for some days, some smoky covering, some particularly different colored growth is observed on that particular food component. And actually that is nothing but a fungus growth. Sometimes you will also see, you have seen that the bread which we eat that gets swollen and that have some black colored markings over that, and that is nothing but due to the fungus. Okay. So if we talk about a fungus, the kingdom fungi, this kingdom fungi is also known as kingdom mycota. It is also known as kingdom mycota. And when we study this kingdom mycota, it is known, this kingdom fungi is also known as what? Kingdom mycota. And this study of fungus is also known as mycology. Okay. Now, when we talk about this fungus, the fungus consists or constitute a unique kingdom of heterotrophic organism. Means this fungus are totally heterotrophic. They are totally dependent on another organism for the food. They are dependent on another organism for the food. Okay, so mainly we talk about them. Here you can see this is the particular classification of the fungi which we are going to study in later part in detail. Okay, and here you will study that there is a particularly larger fungus is that in that you will find a subterranean fungi like Trappes, and in above ground you will find some of the particularly Basidiomycetes and Ascomycetes, which are some of the higher fungi that are known to be seen on the upper part, and then a particular different classification of fungus is found. And this all diagrammatic represent show the different types of fungus. So there are a variety of fungus are found, but all are heterotrophic. So it is known that if we see the variation in way, what in variation is observed in which way. So it is known that they show great diversity in morphology and habitat. What they show? They show a great diversity in morphology and habitat. And this great diversity in morphology and habitat is seen in case of this fungus. Means morphologically they have various shape and they are found in different habitats. You know that when the bread develop a mold or orange rot, it is because of fungus. As you can see in the particularly this is a diagram which is developed on this one. Then the common edible mushroom and toadstool are poisonous, are also a particularly fungus. You know that mushroom is edible one, but actually a poisonous mushroom are known as toadstool. And this poisonous mushroom toadstool are not edible, but these both belong to fungus. I hope you know this. There's a mushroom. Okay. Then sometimes we see that white spots are found on the mustard leaves, and that are also due to the parasitic fungus, that is albuco. Okay, so this is also a due to the fungus. You can see this is the white spot by the particular parasitic fungus, particular albuco, and it is known as a white rust of crucifers. What it is known as? It is known as white rust of crucifers. Crucifers are nothing but belong to the Brassicaceae family. So it is due to the albuco. Some unicellular fungi are also found. The fungi can be unicellular as well as multicellular. The unicellular fungus are known as yeast, and you can see these are the particular diagrammatic view. And this yeast are used to make and bread and beer, and we will study that part also in detail. So this is the bread, you can see beer that also can be made by the yeast. So we will study about that yeast also. Fungi which cause disease in plant and animal. They are able to cause disease in plant and animal. And when we talk about the particular genus Puccinia that cause a wheat rust, means is cause a disease in particularly wheat, that is known as rust. You can see this is a diagrammatically shown a rust which is covered on this particular stems of wheat. Then the life cycle of this Puccinia gramineus, we will study this when we will study Basidiomycetes. Okay, so till now just I am making you to understand what is the particular fungus. We have studied that they have shown extreme diversity, great diversity in the morphology and habitat. In habitat also they are found cosmopolitan. Fungi are cosmopolitan, means they found everywhere. They occur in air, water, soil, on animal and plant. They are found everywhere. So because of this, they are known as cosmopolitan, and they show great diversity in habitat as well. In this, they prefer to grow in warm and humid places. This is one of the important concept that you remember. So many times you will find this in the question. Okay, they prefer to grow in warm and humid places. Okay, and in that also you will find that. Okay. Next to that, if we discuss, we keep food in a refrigerator to prevent the food from the contamination. And that is what it can be bacterial or fungal contamination. But usually from the fungal contamination, because in a refrigerator, the temperature is too low, so this fungus become inactive. They are not able to germinate on this food. With the exception of yeast, which are unicellular, others are known to be multicellular, and in multicellular, fungus are known to be filamentous. You can see filamentous, like if we see the anatomy of this fungus, then you will find a filamentous fungus. When we study their bodies consist of long, slender thread-like structure that is known as hyphae. And the group of this hyphae is known as mycelium C. These are the hyphae. So if there is a particular fungus is growing like this, and this is the body of fungus. Now in this fungus, this body is known as mycelium, and this mycelium of fungus consists of network of hyphae. When we see this hyphae are of two types. This hyphae, you can see this hyphae are of two types. The first one, that is septate hyphae. In septate hyphae, if we see the hyphae in detail, you will find such cross section. Such cross section you will find. You will find these cross walls, and this cross wall are known as septum. So septum is nothing but cross wall in the particular cytoplasm, and due to this nucleus are separated from each other. So this is the particularly a single cell, and the single cell has a one nucleus. So if the single cell has a one nucleus, so this septate hyphae become monokaryotic. Why they become monokaryotic? Because in their hyphae internally cross walls are present, nucleus are separated by cross walls, and one cell has a one nucleus, so monokaryotic hyphae are the nothing but a septate hyphae. But when we talk about sinusotic hyphae, they are aseptate hyphae. In aseptate hyphae, what happens? The cross wall which are found in septate hyphae, the cross wall are absent. And if this cross wall are absent, then the multinucleate hyphae will form. In hyphae, multiple nucleus will be observed. You can see there's a blue color shown here. These are the multiple nucleus, and due to multinucleated hyphae is known as sinusotic hyphae. So this sinusotic hyphae is a term for the multinucleated hyphae. Okay, then okay, I hope you have understood this. I will remove this, then we will discuss the further part. Now after this, you will know that the network of hyphae is known as mycelium, which we have studied thus, and in this hyphae you have to study hyphae. Hyphae can be septate or aseptate. If it is septate, then it is a monokaryotic, and if it is aseptate, then it will be multinucleated, and multinucleated hyphae nanosinocytic that we have discussed. Now, same hyphae are continuous tube filled with multinucleated cytoplasm that is known as sinusotic hyphae that we have discussed. You can see again this particular diagrammatic view. And when we study the hyphae of fungus, we come to know that the hyphae are particularly have a cell wall, and this cell wall are chitinous cell wall. Which type of cell wall? Chitinous cell wall are found in this. Others have a septa or cross wall, and their hyphae are particularly aseptate, and aseptate have a monokaryotic condition. So the cell wall of fungi are composed of chitin and polysaccharide. Chitin is mainly present in case of this. And when we see the fungal cell wall, in fungal cell wall, you can see this type of structure is found. This detailed structure is not required, but you should remember chitin is there, and this chitin is a polymer of N-acetyl glucosamine. Okay, you should remember this, that this chitin is a polymer of NAG, and NAG is nothing but N-acetyl glucosamine. Okay. When we talk about the reserve food material, the glycogen and oil are the reserve food material in case of this particular fungus. Then most of fungi are heterotrophic, and they absorb soluble organic matter from the dead substrate. Hence they are called as saprophytes. They are known as saprophytes because they absorb a particularly soluble organic matter from the dead substrate, and those which absorb this particular dead substrate food from the dead substrate, then they are known as saprophytes. So you can see these are the fungus which are going on the wood logs. So those are dependent on the living plant and animals, they are called as parasite, but which are dependent on the dead one, they are known as saprophytes. So this is about the nutrition. You can see ringworm, athlete's fruit, that all are caused by this fungus. It is known that they can live symbiotically as well. As if you talk about nutrition they can be observed what? Saprophytic. They can be observed a particular parasitic and they can be observed a particular symbiotically in association with the algae, then they are known as lichen. And if they found symbiotically in association with the higher roots, that mycorrhiza. So if you want to define a lichen, lichen is a symbiotic association of algae and fungi. And we will be going to study this lichen in detail in the end part of this chapter. Mycorrhiza is a symbiotic association of fungus with the roots of higher plant, and higher plant here include gymnosperm and angiosperm. So you can see this is the lichen. This is the mycorrhizal diagram. We will study about mycorrhiza also in this lecture, in this chapter in detail. Now from here we will be talking about reproduction, and when we talk about the reproduction in case of this fungus is can be vegetative, asexual and sexual. When we talk about vegetative reproduction, the vegetative reproduction in fungi take place by a means of fragmentation, fission and budding. Fragmentation can occur, fission can occur and budding can occur. So you know that fragmentation, what happens? The body is fragmented into the different parts, and each part has a capacity to give rise to new mycelium. So in fragmentation, what happens? The body get divided into several fragments due to the particularly what we can say any mechanical or environmental pressure. So when the body get divided into fragment, this each fragment has a capacity to form a new mycelium like this. So this is known as fragmentation. Now, another type which is found here, that is binary fission. And you can see here in case of binary fission that the particularly binary fission occur here, and in binary fission the particular cell which having a DNA and cell envelope get divided into two daughter cells. So bi means two, that fission is occurring into the two one. And the third one which is found, this particularly budding, and budding is the what? A development of extra protuberance. Yes, students, here in budding extra protuberance, this is known as bud is developed. See, this is undergoing a fission. Okay, so it is divided into two, but here this is the budding yeast, and in budding yeast what happens? The extra protuberance develop and the nucleus divides and nucleus reach to this extra protuberance, and this will the particularly going to form a new particular species. So in Saccharomyces species you will find the presence of budding, but in Saccharomyces it is known as fission yeast, in which fission is observed. Then from here we are going to talk about some more reproduction strategies. In this we have also discussed the reproduction strategy, that is what particularly we can say vegetative one. And in vegetative we have discussed the binary fission, then we have discussed the fragmentation, and also we have discussed the budding. So in budding also we have discussed that is an unequal cytoplasm division, whereas in fission the equal cytoplasm division occurs. Now here we are going to talk about asexual reproduction. Now when we talk about asexual reproduction in fungi, it is known that, now you may remember some of the point with respect to this. When we talk about fungus are haploid, means they have a single set of organism. And if it is a asexual reproduction, then meiosis is not going to occur. Then mitosis will occur. And through mitosis is produced a spore. So this spore are known as mitospore. Now this spore is produced from the haploid by mitosis, so this spore is also haploid spore. And this mitospore are known as asexual spore. So this in an asexual reproduction, asexual spore are produced in case of fungi. So there are different type of asexual spore. There are different type of asexual spore. So we are going to study here type of asexual spore. Now, when we talk about type of asexual spore, we come to know that there is a zoospore, there is a chlamydia, there is a chlamydospore, there is a oidea. These all different type of spore are found. So first we are going to talk about zoospore. Now, when we talk about the zoospore, this spore is one of the important as in the cell wall is absent. So this particular spore is wall-less. Okay, and if it is wall-less, then we can say it is a naked spore. But this naked spore is formed in favorable condition. It is formed in favorable condition. And this zoospore is a mitospore which is produced within zoosporangia. So it is endogenously produced within zoosporangium. Okay, then flagella may be present or may not be, so they can be motile or non-motile. If they are motile, they can be uniflagellated or biflagellated. They can be uniflagellated or biflagellated. This is a uniflagellated. This is biflagellated. Okay, so this both can be possible. Okay, so zoospore are motile, can be non-motile, but usually they are motile flagellated. So I remove this actually. Just I have written by mistake. They are usually motile. So we remove this non-motile nature, and if motile, they can be uniflagellated or biflagellated. Now here this biflagellated zoospore are also two type. Okay, but there is no need to study in this part. And zoospore is found in, if we talk about the location of zoospore, we can say they are found in Oomycetes class of fungi. There is a one class that is Oomycetes in case of Phycomycetes. In that, this particular zoospore are produced. So this is the first type of spore. After zoospore, we are going to study next spore, that is Conidia. So second type of spore is Conidia, or we can call it conidiospore. Now, when we talk about the particular characteristic of conidiospore, this conidiospore are thin-walled. If they are thin-walled, means obviously they are formed in favorable condition. One more thing is that they are non-motile, means non-flagellated, because for motility flagella is required. Then they are exogenously, means outside, exogenously produced over conidiophore. So these are the important points with respect to conidia. So now when we particularly take an example, it is known that these conidia are found in some of the ascomycetes example, that is penicillium and aspergillus fungi, and this both belong to the ascomycetes class. Now, when we talk about this conidia and when we see the structure of this penicillium in detail, then we see the microscopic structure like this. This type of structure is found in case of this one. Now, this is the hyphae. But this is the hyphae which is going to form a conidia. So this is the stalk of this hyphae. So it is known as conidiophore. And this was the hyphae. Now in conidiophore, the primary branch develop here like this. And over primary branch, you will see the secondary branch. And over secondary branch, you will see the development of flask-like structure like this. And over flask-like structure, you will find a development of a chain of conidia, chain of conidiospore. And in this chain of conidia, you will come to know that, see, this is what is this? This is sterigmata. And the sterigmata are also known as phylides. And over this is the young conidium. Conidium is a singular for the conidia. And this is the old conidium. So when we see young conidium is at the base and old conidium at the tip side, this is known as basipetal arrangement of conidia. And this is found like this in case of penicillium. So this is all about what another asexual spore, that is conidia. Now one of the important statement about this conidia that we have to mark, and that is particularly that is the most common type of spore in case of fungi. I'm writing one statement here. We remember it is a most common type of spore in fungi. You should remember this statement that is important with respect to question. Okay. Now after this, we are going to talk about third type of spore, that is sporangiospore, or we call it a aplanospore. Aplanospore are known as they are non-motile, means they are non-flagellated, they are thin-walled, and they are formed in favorable condition. They are formed in favorable condition. They are non-motile. They are thin-walled, and they are endogenously produced within sporangium. Now it is known that these are aplanospore which are non-motile, and whatever we have studied motile spore, that is zoospore, they are also known as planospore. But these are non-motile, because of this they are known as aplanospore. If we talk about the examples, it is known that these aplanospore are found in zygomycetes fungus. And zygomycetes fungus is a class of fungus in case of phycomycetes. So this is the aplanospore. After aplanospore, next we are going to talk about another type of spore, that is a chlamydospore. And that is a fourth type of spore, that is known as chlamydospore. Now what are chlamydospores? Suppose this is the particular filamentous fungus, and in that one thick walled resting spore develop. Then this thick walled resting spore is known as a chlamydospore. And this thick walled resting spore, it is formed to pass the unfavorable condition. So it is mainly formed in unfavorable condition, and in that this particular fungus is going to pass this unfavorable condition. So because of this, these are also known as perinatic spore. They are also known as perinating spore. Okay, dear student, so this is a chlamydospore. So these are the different particularly type of spore: asexual spore, a mitospore, which are found in case of fungus. Now after this we are going to talk about the sexual life cycle, but in sexual life cycle we'll talk about different stages. Okay, till that we have completed now the asexual reproduction in case of fungi. After asexual reproduction, now we are going to study, these students, a sexual reproduction in fungi. So when we see the sexual reproduction, in this sexual cycle occur in case of fungi, and this sexual reproduction involves three steps. First is plasmogamy, and in plasmogamy, the protoplasm between the two motile or two motile gametes fused, and that is known as plasmogamy. So plasmogamy is the fusion of protoplasm, and that is the first step. After that, next stage will occur, that is a fusion of nucleus, and that is known as karyogamy. After karyogamy, the new meiosis will occur in zygote, and the resulting haploid spore will be produced. And that is shown diagrammatically here. So when we look here in diagrammatic purpose, you can see that here this is the particularly hyphae, and this hyphae produce two type of spore, that is male spore and female spore. Now this spore, particular male gamete and female gamete, sorry, this male gamete and female gamete are going to fused by the protoplasm and the heterokaryotic means dikaryotic condition is formed. After this two nucleus is going to fuse, that is known as karyogamy, and the zygote will be formed, so zygote will be diploid. This hyphae is haploid, so obviously male gamete are haploid. Then the zygote undergo meiosis and produce a spore that are haploid. When the spore germinate, they will again going to form a hyphae. So if I draw a particular diagrammatically, suppose these are the hyphae. This is the hyphae of male fungus and this is the hyphae of female fungus. And we have taken monokaryotic fungus. Now, in this, the protoplasm of this two cell is going to fuse, and that is known as plasmogamy. And when plasmogamy occur, the hyphae will look like this. This round circle are known to be nucleus. So you can see in each cell there is only single nucleus, but in this cell there is a two nucleus. So this is the condition is known as dikaryon condition, and the phase is known as dikaryophase. After dikaryophase, the karyogamy is going to occur, and in karyogamy nucleus is going to fuse. So when this nucleus is going to fuse, this will form a diploid zygote. So after karyogamy, the diploid zygote will be formed. And this zygote undergo meiosis to produce haploid spore. Okay, because it was also haploid, because fungus are haploid. They have a single set of chromosome. Here in dikaryophase, they become N plus N. And after this zygote, they become 2N. Now sometimes in some fungi happens that dikaryophase is not found, and in some dikaryophase is found. So usually in septate fungus, in the fungus which having a septate hyphae or the higher fungus have this dikaryophase. So one more thing that you should remember, that this dikaryophase, I am writing this point here, is present in Ascomycetes and Basidiomycetes. In Ascomycetes, this dikaryophase is short-lived, and in Basidiomycetes, it is long-lived. Whereas this is absent in Phycomycetes, aseptate fungus. So in lower fungi, Phycomycetes, this dikaryophase is not found, but in higher fungi like Asco and Basidio, dikaryophase is found. So in such a way, the sexual reproduction is going to happen. When we talk further, when a fungus reproduces sexually, two haploid hyphae of compatible mating type come together and fuse, as we have seen in this diagram. Yes, we have drawn this diagram and that is shown here also. Then gametangi are going to fuse and zygote and zygospore is formed. We will study that in further detailed diagram. In some fungi, the fusion of two haploid cells immediately result in diploid cell, means dikaryophase will not be observed. If the two haploid cells fuse immediately, dikaryophase will not be found. Now fungi, Asco and basidiomycete is in this the intervening dikaryophase is found, that is two nucleus per cell occur in such condition called a dikaryon, and the phase is known as dikaryophase as we have studied. Later the parental nuclei fuse and the cell become diploid, that is zygote. Now this three steps are involved, but in this the plasmogamy is of different type. Plasmogamy occur by a several mechanism, by a several meanings. So we are going to study the type of plasmogamy, a fusion of protoplasm here.